Hello, everyone. Thank you for joining us today for Molecular Connectivity Online Series, or MCOS. I'm Cindy from New Jersey Institute of Technology. For those who are joining us today for the first time, the Molecular Connectivity Working Group is a community of researchers dedicated to advanced molecular imaging as a tool for studying brain connectivity. We plan this series as a platform to exchange ideas and promote the latest research in this field. You can visit our website, subscribe to our newsletters, or follow us on Twitter or LinkedIn. Before we begin, a quick note. We are recording this session and will upload it to our YouTube channel. To protect our research of the speakers, we kindly ask you not to do a private recording or upload this content to any third-party website. Now I'm honored to introduce today's speaker, Dr. Johannes Gnoerich. Dr. Gnoerich is a clinician scientist at LMU University Hospital in Munich, where his research focuses on PET imaging, neuroinflammations, and biomarker development in neurodegenerative diseases. He has published several key translational studies, notably developing some neurotracers to disentangle the cellular sources of PET signals. By integrating advanced network-based imaging and metabolic connectivity, his work beautifully bridges the gap between single-cell biology and system-level brain alterations. Today, he will be presenting on deciphering the biological correlates of metabolic connectivity in neurodegenerative diseases. Please join us to welcome Dr. Gnoerich.
Thanks for the kind introduction and of course again for the invitation to be part of this series. I'm really intrigued to get to know you guys and also be part and talk a little bit about our recent research. First of all, I will share my screen, right? And
Yes.
maybe get a quick confirmation that you can see everything properly. So you should be able now to see the big screen, right?
Yes, perfect.
Oh, okay, perfect. So nice. Okay, hang on. I just need to. Okay, so yeah, today I would like to talk, as you already mentioned, about metabolic connectivity in neurodegenerative diseases. But more specifically, I want to talk about this readout and what it reflects on the biological level. For this purpose, I will move through these 4 steps. I will first motivate why this question is relevant by discussing what regional FDG PET can and cannot tell us. I will then introduce metabolic connectivity as a way to move beyond regional uptake and towards network level relationships in FDG PET data. And the third part will focus on the preclinical validation. Here I will summarize how our group has established metabolic connectivity in preclinical mouse models. And the next part is also closely linked to that because before we can actually interpret metabolic connectivity biologically, we need to know when the readout is actually technically robust. And at the same time, the biological question defines what actually needs to be validated. So in the second half, I will more or less connect both aspects, the validation and biology, by using preclinical mouse models, cellular tracer uptake, and network level PET analysis. Exactly. So, as you probably are all familiar with, neurodegenerative diseases are characterized by progressive neural loss. They are divided or can be divided by their heterogeneous underlying pathologies, including tau pathology, amyloid pathology, or TDP-43, prion pathology, and alpha-synuclein pathology. Today, we have many PET tracers available that can actually visualize these proteins. For instance, fibrillary amyloid by amyloid PET CT, or in neuroinflammation conditions, we have TSPO PET for microglia or deprenyl PET for astrocytic imaging. Across all these heterogeneous pathologies, FDG PET remains highly relevant because it captures a shared downstream consequence of all of these neurodegenerative diseases, and that is the neuronal loss or the metabolism. It does not tell us which pathology is underlying, but it can tell us where in the brain the metabolism may or might be altered. we can move from, or when we move from human neurodegenerative diseases to preclinical mouse models, the interpretation of FDG PET becomes a little bit more complex. In Alzheimer's disease patients, as outlined here on the left side, FDG PET typically shows a hypometabolism in typical cortical regions, as here the temporal parietal lobe, which is normally interpreted as a marker of neuronal dysfunction or neuronal loss. In contrast, several mouse models, amyloid mouse models, have shown an increased FDG uptake, a so-called hypermetabolism, despite the presence of amyloid pathology or amyloid plaques. And this creates a discrepancy between the human situation and the preclinical mouse model and raises the question of what FDG PET actually measures at the biological level. In the next slides, I will therefore focus more on the sources of the GPET signal and challenge the traditional neuron-centered interpretation of brain glucose uptake. This brings us now to the cellular origins of the FDG PET signal. As I have just mentioned, the classical interpretation of FDG PET has been normally or has been recently been neuron-centered. But the PET signal in general is a composite of the tracer uptake at the cellular level and the underlying cells. This is a study by our group that has shown that microglial activation states can strongly influence glucose uptake and FDG PET alterations in mouse models of neurodegenerative diseases. In general, mouse models of neurodegenerative diseases, they have a really prominent neuroinflammatory component at least. Under resting conditions, microglia may contribute only to a smaller fraction to the total FDG PET signal, but upon activation, the metabolic demand increases and the relative contribution to the FDG PET signal can become much more important. Microglia, however, are not the only player or only larger contributor to FDG PET uptake. Also, astrocytes may play a major role. And this is work that has been shown by Zimmer and colleagues, which provided important evidence that the FDG PET signal is strongly linked to astroglial glutamate transport. More specifically, the activation of astrocytic glutamate transport can trigger glucose uptake in astrocytes. This is biologically plausible because astrocytes are tightly coupled to synaptic activity. They clear glutamate from the synaptic cleft and maintain neurotransmitter homeostasis and support neuronal metabolism. Therefore, part of the FDG PET signal that we often interpret as neuronal activity may actually be mediated through astrocytic responses to neuronal signaling. This is the point where the biological question becomes very important, because if FDG PET is composed of neuronal, microglial, and astroglial components, how can we actually disentangle all these different contributions to the total uptake experimentally? And that's where we have established or developed a method. This is a so-called single-cell radiotracing approach or radio sorting after in vivo injection approach. The principle is quite straightforward. So first, the radio tracer, in this case, of course, FDG, is injected in vivo. The tracer distribution occurs under physiological conditions, and after the initial uptake phase, where we can also perform an FDG PET, for instance, the brain is extracted and dissociated into a single-cell suspension. The cellular debris and red blood cells are removed, and specific cell populations can be labeled and isolated, for example, using a magnetic cell separator or MAX sorter or flow cytometry. Finally, the radioactivity is measured in a gamma counter, and the single sorted cell fractions can be measured. And this allows us actually to quantify how much tracer uptake is attributable to a specific cell population, such as neurons, in this case, the microglia, but also astrocytes and other glial cell populations. The major advantage of this method is that it directly links the PET tracer signal to its cellular sources. So instead of only asking whether a brain region has higher or lower FDG uptake, we can actually ask or answer which cells are responsible for this signal alteration. Using this type of approach, we can move from regional PET to cellular allocation model of FDG uptake when combining the cell-specific uptake and the absolute cell numbers. This present hypothetical model I'm showing here or displaying here used assumptions of cell numbers and estimations of glucose uptake in the intra and extracellular compartments of the interstitium. This slide is supposed to illustrate what the overall FDG signal already in the wild-type mouse looks like. And it is not only the sum of neurons or synapses, but a composition or major composition and contribution is also coming from oligodendrocytes, astrocytes, and also microglia cell, which is even increasing in the A-beta mouse model. This model here is also important for the rest of the talk because it changes how we interpret FDG PET. If cellular allocation changes between healthy and disease states, then also regional FDG uptake will most likely change as well. And this leads us to the next step, because if regional FDG PET is a cellular composite, then also FDG-based metabolic connectivity measures may also reflect more than just the neuronal network organization. Now, Igor is also here in the group, and he's probably really familiar with this graph. Still, I want to quickly introduce the concept of metabolic connectivity, although I suppose that most of you are familiar with it, because metabolic connectivity extends the FDG PET from regional signal intensities to interregional relationships. And then rather than just focusing on only whether a single region shows increased or decreased uptake, it can also show how these patterns are correlated across different brain regions. There are many different methodological approaches to model these relationships. For this talk and most of the data I will present, the most important concept, or the concept we're using, is the correlation-based approach. So if the FDG uptake is correlated between two different brain regions across animals or subjects, this would suggest that there's a metabolic relationship between these regions. So metabolic connectivity has been applied already in several neurodegenerative diseases, for instance, in dementia with Lewy bodies, frontotemporal dementia, Parkinson's disease, and also Alzheimer's disease. And I think what is quite interesting that over the last years, many PET studies incorporating metabolic connectivity have gained increasing clinical relevance. So I just, I think yesterday when I was finalizing the last slide, I was looking in PubMed and typed in metabolic connectivity to check for the results. And I think it's quite interesting that in the last 2 years, so 24 and 25, there was a significant increase compared to the years before. I don't know why we had this plateau there in 21, 22, or 23, maybe because of many COVID studies that could have been of more interest. But in the last 2 years, again, metabolic connectivity has risen significantly. And this now raises for us a critical biological question: what actually drives these FDG-based network patterns? Are they purely neuronal, or can also neuroinflammatory processes reshape metabolic connectivity? The first study I want to present, here we focused on microglia particularly and neuroinflammatory conditions. More specifically, we used two different and opposing microglial activation states. So here I want to focus on TREM2 and granulin. These are two genes that are under physiological conditions responsible for the not only the neuroinflammation, but also for the response to neuroinflammation. However, they have opposite effects when they are actually knocked out. So in these mouse models we used here, in this study, we actually used loss of function knockout mouse models where the loss of TREM2 and the loss of granulin led in the end to neurodegeneration. Yeah? So the loss of function of TREM2 leads to a homeostatic signature, so the microglia actually become inactive. And in the progranulin loss of function mouse model, they obtain a disease signature where they are really activated. Now, given that immune cells can strongly the influence of the FDG PET signal in general, this creates an interesting paradox, because here we have opposing microglial activation states. Both models show similar hypometabolic pattern, as you can see on the left side. So this is an FDG PET, always compared to wild types in all of these mouse models. So in the granulin knockout mouse model, as well as in the TREM2 knockout mouse models, and as a crossover mouse, double knockout mouse model, we have a hypometabolism, although we have opposing microglial activation states. Therefore, we ask whether network analysis of FDG PET could reveal functional differences between these two models that remain hidden at the level of regional or global uptake alone. To address this question, we translated FDG PET from regional uptake into metabolic connectivity maps, and this allowed us or enabled us to test whether these network level relationships differ between TREM2 knockout mouse models or granulin knockout mouse models. For this purpose, we extracted uptake values from 24 cortical and subcortical volumes of interest as depicted here on the left side. We calculated interregional Pearson correlation coefficients and applied Fisher's Z transformation, and the resulting correlation matrix was then projected into a 3D mouse brain to visualize the regional nodes and connections. The first important observation, I want you to first look at the axial and corona FDG PET image. We could confirm the previous results. So there's a hypometabolism in the TREM2 knockout mouse model and the granulin knockout mouse model compared to the wild types. So based on the regional FDG uptake alone, these models appear relatively similar. They all show hypometabolism, right? However, when we now look at the metabolic connectivity, we do see a very different picture. So the granulin knockout mouse models showed a significantly or marked increase in metabolic connectivity, particularly in the cortical networks, but also in the hippocampus. This increase was not observed to the same extent in the TREM2 knockout mouse model, but also not in the double knockout mouse model. Here, the deficiency was largely suppressed also. The key message of this slide is that regional FDG uptake and metabolic connectivity can actually dissociate. So they don't have to be going the same trajectory, they can dissociate. Similar levels of hypometabolism do not necessarily imply similar network organization, and metabolic connectivity therefore provides information that is not captured by regional FDG PET alone. To test to what extent microglia themselves contribute to metabolic connectivity, we used pharmacological microglia depletion. We administered PLX5622. This is a CSF1R inhibitor that allows us to reduce the microglial population in the brain by approximately 96 to 99%. So basically, there's no more microglia left. And after microglia depletion in these two independent cohorts with two different ages, so this first cohort was 6 months old, the second cohort was 9 months old, the administration of the plexine was about 1 month. We compared the metabolic connectivity patterns in all of these groups. And what we found was quite impressive. So there was a markedly reduced metabolic connectivity or ICCs in the administered or the treated group compared to the vehicle group, where basically no significant changes occurred during this 1 month of period. So this information, together with the knockout mouse model data, supports the idea that metabolic connectivity is sensitive to microglial states, both at the presence and the activation status of microglia, which shape the FDG PET network relationships. And now to further elucidate how FDG uptake is allocated between neurons, microglia, and astrocytes, in these mouse models, we use the single-cell radiotracing approach again, which I have just introduced before. So here the key observation is that the single-cell radiotracing data shows that the FDG uptake per neuron is strongly reduced in the granulin knockout mouse model. That implies that the lower conventional FDG PET signal in this granulin knockout mouse model compared to the wild-type mice is likely driven by reduced neuronal FDG uptake. However, when we look at the microglia, the single-cell uptake was not strongly increased in the granulin knockout mouse model compared to the wild types. But the CD11b immunoplot showed significantly higher CD11b expression. We know that CD11b expression per individual microglial cell is unchanged. So this indicates that the increased the increased CD11b here indicates a microglia abundance or proliferation of microglia. We have a higher number of microglial cells within the brain of granulin knockout mouse models compared to wild types. So basically, this tells us that not only we have more microglial cells, we have also this reduced neuronal uptake, and this actually allows us to calculate the total contributions to the FDG PET signal we get a new relative allocation of FDG PET signal, which is shifted towards the neurons, here indicated as blue, towards the microglia or the astroglial cell fraction or compartment. The next important step was to validate for us how robust is metabolic connectivity actually as a preclinical PET readout. Because the key methodological question for us was how many animals do we actually need to obtain stable and interpretable results or connectivity estimates. Also, if we think about the 3R principles, we always want to reduce the number of animals needed, but of course we need sufficient animals to get robust results. So to address this, we analyzed P301S mice. This is a mouse model of 4 repeat tau and wild-type mice, and recalculated the ICC matrices at different sample sizes and evaluated 2 aspects: the stability of the matrices using the root mean square error here on the left side, and the ability to detect group differences using COINS-D. The threshold was around 12 miles, and that emerged because at this point ICC estimates became sufficiently stable, and the SUVR based metabolic connectivity, so using the global mean as a reference region, so the SUVR reached a meaningful effect size for the P301S mice compared to the wild-type mice. Many previous studies have shown that there can be alterations to cortical, but also global mean or whole brain FDG PET uptake in animals that were injected awake compared to animals that were injected under anesthesia. We therefore wanted to test whether the conscious state during FDG uptake also affects metabolic connectivity. For this, we performed a head-to-head comparison in 11 6-month-old female wild-type mice. Each mouse was scanned twice within 1 week interval, once after FDG injection in the awake state and once after injection under isoflurane anesthesia. Although anesthesia could theoretically alter metabolic patterns, global mean normalized regional FDG uptake was largely comparable between both conditions, and more importantly, the resulting connectivity matrices showed similar network patterns with a strong overall correlation between ICCs from both conditions. So in our setup, at least, anesthesia did not significantly alter metabolic connectivity, supporting the robustness of the readout under standardized experimental conditions. The next validation step for us was the inter-scanner comparability. This is relevant because preclinical PET data are often acquired on different scanner systems, which can differ in spatial resolution, sensitivity, or also reconstruction procedures, for instance. For reproducibility analysis, we calculated ICCs, as I have shown before, for APPPS1, beta amyloid mice, and also age-matched wild-type mice. Images were acquired on 2 different scanners, the Siemens Invion dedicated PET scanner and a Mediso scanner, which is actually a PET-MR scanner. And based on the SUVR data from both scanners and the connectivity matrices, they showed a strong quantitative agreement when correlating all ICCs for both the APPPS1 mice and the wild-type mice. And the quantitative comparison of intraneocortical ICCs, no significant differences between the PET scanner and the PET-MR scanner were evident for both mouse models. After validating metabolic connectivity in disease models, we next wanted to understand whether similar principles also apply during the healthy aging. So here we analyzed mice from 3 to 18 months of age. We therefore combined FDG PET with single-cell radiotracing and determined how different cell types contribute to the FDG PET signal as a function of age. As the first finding, here you can see the coronal and the axial PET images, FDG PET images. We showed that the cerebral FDG PET uptake increased linearly with age in all of these wild-type mice significantly. Next, we found that microglia and astrocytes showed a decrease of FDG uptake from young to intermediate ages, followed by a recovery toward the late ages. Contrarily, FDG uptake in neurons indicated a distinct increase in glucose uptake from young to intermediate ages, followed by a subsequent strong decrease toward the late ages. So In summary, neuronal FDG uptake explains the increase in FDG PET signal from young to intermediate ages, whereas the FDG PET signal increases from intermediate to late ages were mostly explained by astroglial glucose uptake. And astroglial is a summary of astrocytes and microglia. We used a linear equation-based allocation model that integrated cell type-specific uptake of these three cell types, age-related changes in cell numbers, and the whole brain FDG PET SUV. This analysis suggested that astroglia contribute more strongly to age dependent FDG uptake or FDG PET signals than neurons across all ages studied, although in the medium or intermediate age, we do see a significant proportion also from coming from the neurons. To better summarize the balance between glial, so astrocytes and microglia, and neuronal FDG uptake, we calculated the so-called FDG glia to neuron ratio. This ratio reflects how much of the FDG signal is allocated to glial cells in relation to neurons, basically. Interestingly, this ratio followed also a U-shaped trajectory across age, which means that glial contributions was relatively higher in young mice, decreased at intermediate ages, and was again higher in the older mice. To test the associations now between metabolic connectivity and cell type specific FDG uptake, during aging, we derived metabolic connectivity patterns from almost 200 wild-type mice. We defined in total 21 regions based on the Mar-Ben-Wisted Merion Atlas with slight modifications according to our previous work, and calculated again interregional correlation coefficients and interregional FDG uptake correlations across age groups. Also here, we analyzed metabolic connectivity in 9 groups with increasing age and observed an initial decrease in the cortical regions. So the cortical regions are here the red boxes in the bottom right, and the subcortical regions are the ones on the top left side, more or less. And in the cortical regions, we observed this increased towards late ages after a decrease from the younger to intermediate ages. While in the subcortical network, metabolic connectivity remained largely stable and did not show any significant age-related changes. This cortical U-shaped pattern, here represented by the black line, is intriguing because it resembles the age-dependent shift we observed also in the cellular FDG allocation, which is here represented as the glia to neuron ratio by the red line. So both the cortical metabolic connectivity and the FDG glia to neuron ratio developed as line trajectories over time. In other words, age groups with a high relative glial contribution to the FDG signal or FDG uptake also show higher cortical metabolic connectivity. We hypothesized that the cortex undergoes more pronounced age-related changes in comparison to the subcortex, including reductions of neuronal density, synaptic connections, but also, or this goes also in line with glial immune cell activation, which may all contribute to the alterations here in metabolic connectivity. In our ongoing work, so this is work that has not yet been published, is still in preparation, we take this approach one step further and directly combine FDG PET, metabolic connectivity, and single-cell radiotracing, as well as single-cell transcriptomics in the same framework in the same animals. We are comparing wild-type mice and APP NLGF mice, which are also amyloid mouse models. And based on this network perspective, we are dividing the brain into a forebrain and hindbrain compartment, and can now analyze both compartments separately. First at the PET level, of course, but of course also in regards to single-cell radiotracing and single-cell RNA sequencing. The overall idea is to ask whether these regional network alterations that we observe in FDG PET can be linked to compartment-specific cellular FDG uptake and cell type-specific transcriptional changes. So as a first step, so here you can see actually the distinct brain regions that are allocated towards the hindbrain and the forebrain. So the forebrain is mostly it's mostly integrating cortical brain regions. And here we first tested for the association between the hindbrain and the forebrain, if we consider both regions as one single region. And here, this simplified correlation analysis showed a quite nice, significant negative correlation in this case. And importantly, this is only the starting point, of course. The next question was for us whether this relationship that we already witnessed here can also be reflected on the level of metabolic connectivity. So similar to the approaches I have presented before, we looked more specifically now on the subregional metabolic connectivities between the forebrain and hindbrain regions. So these are the regions that are here highlighted by the black boxes. In the APP and LGF mice, in the wild-type mice, we had a higher number of significant correlations in the APP and LGF mice compared to the wild-type mice. But also when we compare the overall forebrain and hindbrain connectivity between the two groups, the APP mouse model showed significantly higher connectivity than the wild-type mice. So this slide provides the network level rationale for the next steps. So if the forebrain and hindbrain connectivity is altered in the APP model, we should ask whether this difference is also reflected at the cellular level. So this is why we then separated forebrain and hindbrain, not only for the PET analysis, but also for the single-cell radiotracing to test whether these compartments here we want to test for the underlying specific cellular FDG uptake in these compartments and how they can lead to these altered network relationships. When analyzing both parts of the brain separately, so here forebrain, hindbrain, this is the summary, the whole brain, we found that overall the APP mouse model showed higher cellular FDG uptake in cerebral glial populations, including the microglia, astrocytes, and oligodendrocytes. In contrast, however, the neurons did not show an increase, but rather a trend towards a decrease of single-cell uptake. Next, we tested for the association between the cellular uptake in the forebrain and the hindbrain in the APP. So here the APP and LGF mice are highlighted in red. The wild-type mice are highlighted in black. The APP mice, here we observed the significant forebrain-hindbrain correlations across all analyzed cell types, which in the wild-type mice only reached significance in the microglia. I guess that's currently the problem because of the limited sample size in this preliminary analysis. Similarly, in the next analysis, we related the regional forebrain and hindbrain correlations. So the FDG PET uptake was correlated with the single-cell uptake. And here in wild-type mice, again highlighted in black, these associations showed similar tendencies, so similar positive tendencies, but also again did not reach statistical significance. Again, possibly due to the limited number of animals, but also due to lower dynamic range of cellular uptake, possibly. Therefore, we are currently still increasing the sample size to improve statistical power, of course. As a final step, we are extending this workflow to the single-cell RNA sequencing level. So the idea, the overarching idea, is to identify cell type specific transcriptional changes that are associated with altered metabolic connectivity and cellular FDG uptake. On the right side, in this plot here, you can see one preliminary example where we have already started this analysis. So here we compare APP and LGF mice versus wild-type mice, the expression levels in these mice shown for the forebrain and astrocytes. So we have this information not only for the forebrain, we have it for the hindbrain and for all different kinds of cells. So also microglia neurons, etc. And not only one population of neurons, but the inhibitory neurons and so on. And you can already visually identify some genes that are stronger expressed in APP mice compared to wild-type mice. And we, for this analysis, are particularly interested in metabolic gene programs that are related to glucose transport and glycolysis, as they may provide a molecular link between the altered cellular FDG uptake and the PET-derived network phenotypes. So to summarize already, I would take away three key points or three main points. So first of all, FDG PET is highly sensitive, but of course its signal is cellularly mixed. Metabolic connectivity adds a network level to FDG PET, but yeah, this network level still depends on the biology of the underlying tracer signal. We have learned that this data, or that at least our data suggests that glial cells, especially microglia, but also astrocytes, can substantially shape the FDG-based metabolic networks. And the central message is here that metabolic connectivity is not only a network readout, but also a biologically modulated PET signal shaped by cellular FDG allocation. And yeah, I first of all thank you very much, of course, for coming and listening. I'm looking forward to the discussion, to any open questions, also any questions that occur maybe during the next days. So whenever you come across one of the works we have done, just feel free to approach me, to email me anytime, and I hope yeah, that I can address all of your questions. Yeah, thank you very much.
Thanks a lot. It was a great talk. A lot of experiments shown, a lot of results. So we can open the discussion. If you have any questions, you can either raise your hand and ask them live, or you can write them in the chat. Igor, okay, you're raising your hand analogically. Yes, it's
Yes,
valid.
I'm an old guy, you know. Yeah, very impressive data, Johannes. Thanks a lot. Have a basic question unrelated to connectivity to understand the nature of the FDG signal per se. So you presented a few studies, preclinical studies. So to sum up, what is the extent or the proportion of the FDG signal which is explained by microglia in adult healthy mice? And what do you think, how good is this value translatable to human?
Yeah, thanks, Igor, for the really interesting question. I mean, the translation is here a really important part, right? Because we have to consider many aspects. We have different cell numbers, also proportional cell numbers in the human brain compared to a mouse brain. So in the mice, all these studies here performed in mice show this large contribution by glial cells, so astrocytes and microglia, right? But we know that in the mouse, there are much more glial cells compared to neurons than in the human brain. Us humans have more neurons than compared to the in relation to the astroglial cells than mice. So this is a lack for the translation we need to address. Of course, we need to keep this in mind when looking at these results. Now you asked for absolute contribution of the glial cells. This is really difficult, right? Because this we are now improving the methodology. We are now able to capture 4 different cell types at the same time for the single cell radiotracing approaches. Also, these models are conceptual, right? We cannot measure the FDG uptake in all cells currently simultaneously. But we are getting there step by step. Now we are measuring the largest 4 cell populations, which also now includes the oligodendrocytes. I hope in the future, because we are this procedure, the single cell radiotracing procedure, has one large limitation, which is that because of the mechanical procedure, you lose some of the synapses. Yeah? And of course, also the synapses have a large contribution to the FDG PET signal, which therefore, I guess all this neuronal data we show you here underestimate actually the FDG PET contribution, even in the preclinical setting. So I guess it would be even higher. So currently, we only have these estimation models, but in the future, maybe we can answer your question definitely. I hope so, yeah.
Okay, because you know I'm a physician, and for me it is also interesting what we see in FDG PET every day. Yeah. It's real neural activity, and my last observation is, you know, there are many studies reporting alterations in microglia, in, for example, in psychosis or in affective disorders in human, also including TSPO traces. But so far, we haven't found any alterations, at least consistently, in patients with psychosis and depression, which makes me a bit hope that what we see in FDG PET is in fact mostly neural activity or astroglial, but not microglia, which is much more dynamic, you know, and it would be yeah, I would be concerned if it would contribute to the signal very much. I cannot say significantly. You know what I mean. So from my clinical observation, yeah, I would say I don't think so, but I don't know. Yeah.
But maybe, you probably also know the studies, right? In early Alzheimer dementia, MCI, right? It's a really, really early Alzheimer's dementia where we do actually see hypermetabolism also in the human setting, right? So this is also can be hyperactivity, but can also be the astrocytes that actually contribute to this increased signal. So it is possible to somehow delineate that. Maybe we need more head-to-head comparisons with other tracers. I mean, we now have the deprenyl tracer available, right, with astrocytic activity, which we measure a lot. And also, as you mentioned, the TSBO tracer, maybe if you have more of these head-to-head comparisons, one patient with 2 or 3 different tracers, we can disentangle this also in the human more. Yeah, but this will be open for discussion.
Neurodegeneration is different because there is a cell death, right, or let's say neural injury, and this has not been observed in affective disorders or psychotic disorders. But microglia activation or let's say alterations in general have been observed. You know, so this is discrepancy. But FDG PET, as you know, doesn't have any value in diagnosis of psychotic or depressive or affective disorders.
We also barely actually perform FDG PET with these indications. Yeah, barely, to be honest.
Okay. Thank you very much.
Thanks you.
Other questions? If not, we have a question
in the chat.
Yes.
Okay.
So by Pratik, so given that the white matter region of the brain has more glial cells than the neuron cell bodies, have you ever analyzed the white matter regions of the brain for FDG uptake?
So in vivo uptake. So yeah, thanks for the question. So to be honest, when we have to differentiate now again between the preclinical and the clinical data, I guess. The mouse brain is really small, right? But you can still disentangle some regions because when we think about the human brain atlas, brainstorm atlas, we have 246 regions, and this Marblin Myrion atlas, we use like tens of these regions to still be able to actually differentiate these regions. But we do see some differences, of course, also in the white matter compared to the gray matter. Also for the connectivity analysis, we mainly focused on the cortical regions in the data that I've presented before. Just because we didn't see that many, we didn't find too many interesting results in the subcortex, but maybe it's worthwhile to look more deeply into it. And what we're doing now with the separation of the fore and hindbrain, we are actually able to now go more in detail into this question, and maybe hopefully also get more information than in the past, because in the past, to be honest, we neglected the white matter more or less. But I guess that can be of particularly interesting now when we have this data. But thanks for the question.
Yes, thank you for the question. So I think the more general is that in fMRI, there is now a trend to study the white matter. They are applying network analysis and find organized kind of networks in white matter. So it's the question is like how to interpret this? How do you have any? Of course, those studies are mostly on human fMRI data, and do you have any insight like how to interpret it, or do you believe those kind of results?
In the white matter?
Yeah, in white matter. For example, if you do any graph theory analysis or clustering, you see the kind of a highly structured networks.
Yeah, I'm not an expert on fMRI, to be honest. I mean, we do have the preclinical PET MRI, but the quality of the MRI is not sufficient to perform any connectivity analysis, so MRI-based connectivity analysis. In Munich, we do have a PET MRI scanner, but Igor is a host of it, so it's at the TUM in Munich, so we don't have the direct access to it. So maybe Igor can answer this question. Maybe you guys have looked into correlations between PET and MRI. I don't know. I don't have the answer for that question. I'm afraid.
Johannes, you are always welcome to use the PET MRI system in Munich.
Yeah, thanks.
At TUM. No, we haven't looked into white matter networks. Well, I'm skeptic in general about functional networks from fMRI, even from gray matter and in white matter. Yeah.
It's too much.
Yes, too much.
All right. Other questions? Else, I have a question. Yeah, otherwise.
Ask a question on terminology. So as far as I know, you use the term so far in your publications, synchronicity or something. Do you put any value on the choice of the terminology? So it is for you should have a biological underpinning, or it doesn't really matter. So it's important just to indicate that there is some interaction between brain regions.
Yeah, thanks for the question. I was actually, because I think you were referring to the study in molecular neurodegeneration, which actually, where we're looking at TSPO PET signals. I was thinking about integrating it, but I thought it was too much information. Because for the others, maybe for context, we also looked for microglial connectivity, basically, in which we named synchronicity. And in this study, which is kind of interesting, we also tried to apply an individualized connectivity approach, because all these results I have just demonstrated are on a group level basis, right? But I think in the future might be also interesting to have single level based connectivity results. Of course, you need a reference, a standard, a healthy control group where you can relate your results to. But in the end, it could be really interesting. And we haven't done this so far here with the FDG PET data. And the terminology, I think, it was just introduced because, there was different tracer. The background is different because we're now looking only at the microglia, because TSPO is mostly microglia specific, right? And that's why we try to introduce a different term, but not because we believe it's a different whole different story. The concept is similar. It's just a more specific tracer compared to FDG, and it's looking at the microglia, and the microglia are more or less synchronized. But the results are similar to FDG, and this supports our findings that microglia are a strong driver of these connections now.
Thank you.
I have a question about, the methodology, let's say, because if I understand correctly, so this single cell assessment of contribution. So you extract some single cells from different brain regions, or how does it work?
So this is what we're doing right now. We have these two compartments. So basically, you have the whole brain, the whole mouse brain. It's really small, actually, and then you can by a cell suspension, you get all the single cells, and then you can sort them by max sorting, so magnetic sorting or fluorescence sorting, and then you get this cell population. So you have one fraction which is only microglia, one fraction is only astrocytes. And then in the cell counter, you can count how many cells are inside the cell fraction, and you can calculate the uptake. And then let's say you have 10 becquerel of uptake, and you have 1 million cells, so you can actually how many what is the uptake per single cell in this cell compartment in this
and these cells are taken from the same. So I maybe I missed it again, but from the same region or from different samples all over the brain.
So this is what you can basically choose. You have to be so the whole procedure. You saw all the minutes, right? The procedure takes the whole day. So this is really, really exhausting experience. You start in the morning and end in the night, basically, or in the evening. So you have to have a sufficient yield of activity to be able to measure in the end is the activity in a single cell, right? So you have to be not only fast, but you have to have enough cells and also enough activity. So this is one limitation. So we cannot just look at one small brain region. We could pool many different mice of the same genotype and take the same brain region of all of these mice. But this is one possibility, but the other possibility is to take a larger part of the brain, right? So this is a limitation. We cannot just put one cell in the morning, incubate one cell, and then get the activity of one cell. That's not possible. So this is why we take the whole brain currently, but now we're looking at two different compartments, so the forebrain and the hindbrain, and this is still sufficient. But I think if we would get any smaller, if we would take, for example, just the hippocampus, they would be too small to gain any results from that.
Because my question was like, so what you say about the contribution of different cell types to FDG is a sort of average over the whole brain, or a big part of the brain, or in the last example, hindbrain and forebrain. But I'm wondering, like, if you could look at the variability across brain regions, would you expect this relationship between FDG and different cell types to be constant, or different regions might have a different?
Yeah, that's a great question. Probably will not be constant. I did not include, because we have a third mouse model, which is not included here, which has a pathology, particularly in the brainstem and the cerebellum. This is really interesting because also in the regional FDG PET, you do see differences, of course, which are regionally highlighted. But when you look at the connectivity, this is even more interesting. I did not show the results here because they are more or less still confidential, but you will see them soon enough. And this is, of course, more interesting if you have a global pathology. Also, these amyloid mouse models, they have more or less a global pathology. So there you can more or less take the whole brain, right? But if you have this focal spots, I think this can tell us a whole different story. But for now, I agree with you. Yeah, you need to be more specific and more precise in order to delineate the contributions, of course. Yeah.
All right. We have a question from Mathieu.
Hi, everyone. So thank you first for the very impressive presentation. And I have a little question, but more on the engineering side. When you did your comparison between the Inveon micropet and the Mediso micropet, did you optimize the reconstruction algorithm and parameters? It's a very pragmatic question because 2 days ago we had an Inveon scan in our labs and we are changing. So it's a pragmatic question.
Yeah, thanks for your question. Yeah, we more or less harmonized them. We used a Phantom for that. We also have another study, triple scanner study, where we compared, because we actually have 3 preclinical devices. We also have a PET-CT from Mediso and tried to harmonize all of these 3 PET scanners. So they were more or less harmonized. Because also for us, we want to scan many mice in all of these scanners and have comparable results. So we more or less have to normalize them. So yeah. Although, of course, you have to consider, right? The dedicated PET doesn't have attenuation correction. So this is the first limitation, the PET MR. Okay, so MRI attenuation correction is also a little bit tricky. So you cannot have the perfect harmonization between the scanners, but more or less with filtering, so we put a Gaussian filter on it, and more or less in the end, you get the same resolution. That's more or less okay, I guess. Yeah. I know.
Okay.
How are you guys handling it?
I think we will use the same method as you, using a Phantom and try to optimize the coefficient recovery and all that turns and the signals to noise ratio to obtain yet some comparable performance. But yes, we have the same problem because with our Inveon, we didn't have CT. So the new one, we have one. So yes, it's a problem with attenuation correction and all this stuff, but I think we will manage it. Thank you.
Yeah, good luck. Yeah.
Other questions? Xin?
Hi. I have a question. I think that you show this aging effect is really beautiful. It's a U shape, but I remember is that a kind of comparable with human, because usually with his human connectivity or any measure, it's kind of inverted U shape, right? And peak at middle age and decrease at connectivity or other measurements. And how do you think that your results are comparable with humans or?
Yeah, thanks for the question. I think in general, if you go deeper into biology, from fetus to an adult, you will have many U shapes, independently if they are inverse or not inverse, but you will see many of these U-shaped. I mean, even tau pathology, I think some here are also involved in tau scans. We have a high tau. I think there are many reasonings for U-shapes. And here we see this inverse U-shape. I think it's mainly driven because, as also Igor asked before, we have more glial cells, of course, that actually are contributing to this U-shape in the younger ages and the really old ages due to neuroinflammation. And in the human brain, in the older ages, the neurons, which are more pronounced, so neural loss, you will see will have a larger contribution, and so the U-shape will be going down again. Also, the trajectory will be going down again. So that's the reason, I suppose.
Another quick question. We have 2 minutes. Else I have one, because then, okay, we spoke about aging, we spoke about the different scanners. I have a question about the anesthesia part, because so if I understand correctly, you show that there is no difference metabolic connectivity. Okay, I mean,
Yeah,
how?
no larger difference. Let's say no larger difference. Yeah, no significant. Of course, it's at 11 miles, so you can still improve the methodology. You can optimize this. I agree, but I think if you go into detail, you will find some differences. But I mean, for this larger group based analysis, I think it's okay to put anesthesia into the mice in order to comply with the 3R principles, in order to not put too much harm. And if you compare the mice anyway, I think it's for us it was just to prove that we can perform the experiments under anesthesia.
Yeah, because I mean, this is very useful to know, right?
Yeah.
Because if you can do it, it simplifies a lot the acquisition, I guess. So the point is that there are no large effects. So as long as you look at large differences for whatever phenomena, it will be fine to use anesthesia.
On metabolic connectivity or even on FDG uptake?
We looked at both, right? I think, and if you look at the regional uptakes, there were some regions that were significant. I mean, if you test for many regions, you will always find one region that is only outlying. But if we looked at metabolic connectivity in particular, there were no significant differences.
Metabolic is for me acceptable, but I would expect rather global decrease, or at least subcortical thalamus.
Exactly. Yeah.
Decrease in FDG uptake.
Yeah.
Cool.
It's very surprising for me because I work with disorders of consciousness, and also we have anesthesia, and we are used to think there is both things create huge effects on anything in the brain. But okay, maybe the mice are built different. All right. Any other question? Okay. If not, Ksenia, you want to give the last remarks?
You can go ahead.
All right. So, well, thanks a lot for joining. Thanks a lot to Johannes for this super comprehensive presentation. It was super interesting, and I'm really looking forward for the next studies. Very, very exciting. When are we meeting again, Ksenia? When is the next MCOS?
We
after
do
summer?
not have a plan. We do not have a plan in the summer. We are planning for the September, but we now,
So we see each other in September.
Yeah. Yes.
Perfect.
Hopefully,
right.
yeah.
Of course. All right. So enjoy summer and see you in September for the next MCOS. All right. Thanks a lot.
Hello, everyone.
Thanks, guys, for having me.
Thank you.
Bye-bye.
Thank you very much.